Hey, everybody. Happy Valentine's Day and welcome back to Winter College Week. Today, as part of the Miami University Alumni Association's virtual Winter College webinar series, we'll be presenting Magic Microbes with Dr. Andrew Jones, who is Associate Professor in the Department of Chemical, Paper, and Biomedical Engineering. Andrew has gone out of his way today to wear a mushroom shirt, so I hope that he gets lots of applause in the chat for being a fun guy. I'm going to really lean into puns this whole time, Andrew. So I hope you'll join me on that journey. Right before we went live, Andrew and I confirmed that we're both native Georgians. He did his undergraduate and master's work at Mercer University in Macon, Georgia, and then completed his PhD in, wow, I don't know how to say the name of your graduate institution.
Rensselaer.
Thank you. He's been at Miami since 2017, and his research focuses on genetically engineering microorganisms for high-value chemical productions. And today he's going to enlighten us all about magic microbes. So I'm going to just turn it over to you, Andrew, before I mispronounce anything else. And I'll be here gathering questions from the audience. Please remember that you can use the Ask a Question button to submit those queries live, and we'll get to as many of them as we can as we move on through the presentation today. Thanks for being here.
Yeah. Thank you for the introduction, and I'm really happy to be here, share a little bit about what my lab's been working on the last 5, 6, 7 years. So we'll get started. My goal for this talk is really to show you what I've been doing, the way that I tackle science, and show you about some of the cool outcomes of my research, and also lay a groundwork for what I see as like maybe the future of mental health treatment or psychedelic medicine. I want to start out by acknowledging kind of my team, right? Most of the work, nearly all of the work that you're going to see today is not things that I personally did, but I led a team of really talented scientists and students to kind of help me achieve some of this work. So, and I'll highlight them throughout the talk, right? I'll put their kind of the lead author for some of the work. I'll put their kind of headshot on those slides. But really, it's this group of people, a fantastic, hardworking group that's kind of doing the heavy lifting. So I want to make sure that introduce them and highlight them kind of first and foremost. As we dive straight in, kind of a path for my talk, right? I first want to tell you a little bit about my field, right? Metabolic engineering is kind of the subfield where my research kind of lives. So I want to tell you a little bit about that field and what that is and how we kind of tackle problems as a field. I then want to transition and start thinking about psychedelic medicine, right? Telling you about psychedelics and why they're exciting, why I'm kind of pushing for research in that area. The big bulk of my talk is going to be thinking about how do we produce these psychedelics, primarily in bacteria. I work mainly in E. coli. And so then after I tell you about some of the work that I've done, I'm going to pivot to the work of some of my collaborators, right? So I really focus on the biosynthesis of these drugs, and then I make those available to researchers across the country that are starting to dive in and study how these things work, right? We start with animal models and kind of ex vivo types of models. And we're learning a little bit more about these psychedelics and how they interact with biology, right? And then I want to talk about the future. All right, so diving straight into metabolic engineering. Right here, I've shown you kind of a piece of the metabolic map for E. coli, right? This is a lot of detail. You're not supposed to be able to pull out all the things, all the specific pieces here, right? But if you think about all the chemical structures in between all of the different enzymatic reactions, right? All the arrows are enzymatic reactions. But what you should take away is that it's really complex, right? It's really interconnected, right? And that this idea of like, if you remember the movie Butterfly Effect, where small changes over here can kind of affect bigger changes over there. That's kind of at play, right? We can manipulate things on one side of metabolism and it can have kind of ripple effects through how these microbes live and function. And that's really what we're trying to do. We're trying to add pieces and take pieces away to help improve kind of the metabolism. But let's say for a minute that we zoom in, right? And we have this metabolic map, this metabolic pathway here, where we're starting with glucose kind of in the top center. We're trying to feed glucose to whatever organism we're working with. And let's say in the end, we're trying to make butanol, right? That's a kind of drop-in biofuel that would be really good to be able to not have to make that from, let's say, petrochemical sources, right? Thinking about sustainability, right? So if I wanted to, as a metabolic engineer, if I wanted to manipulate the genetics of this microbe to enable it to better produce butanol from glucose, right? A common first step would be to knock out competing pathways, right? So I would take out, physically remove the genes from the genome of this organism that are responsible for this kind of side path towards ethanol or this side path towards acetone, right? And that's going to better channel flux towards the direction where I want that kind of metabolite to go. Right. And then I could overexpress, or I could add extra copies of the genes that are in the direction that I want that flux to travel. Right. So I can kind of, I don't know, that's thinking about, if you think about the road analogy, right, it'd be the X's would be the equivalent of blocking off kind of side streets, right? And the green arrows would be like taking your 2-lane road and making it a 6-lane highway, right? That's kind of the approach that we take. But it's not quite that simple, right? This will get you a lot of the way there. It'll show you some improvement, but it won't get you to industrial level kind of efficiencies that you need for production, right? Because things are complicated, right? The pathway you're looking at is not isolated, right? There are metabolites that get used in many other pathways for many other different purposes in the cells, right? There's balances of cofactors that are used across the board, right? Thinking about some of these steps require energy cofactors or regenerate energy cofactors. And these others have reducing equivalents, right? And these are compounds that are used for enzymes all across metabolism. And if your pathway disrupts the balance of those compounds, it can really throw havoc into kind of how your cells live, survive, and ultimately produce the product you want. But then it gets even more complicated, right? Because sometimes we need to vary the different expression levels to kind of balance all of these systems, right? So we don't need everything overexpressed at the same level, right? And so I change the size of some of those arrows to say maybe some enzymes need to have a higher expression, some need a lower, to kind of properly balance all of these kind of considerations. And then we can toss in a lot of other things that are kind of fundamentally thought of as metabolic burden, right? Things that are going to interact with your pathway or the way your cells are going to manipulate, right? So there's lots of considerations that we have. Oftentimes it's more of a black box. We don't really know what we're trying to do. Like we have an idea, but like when we change one thing, it's like we can't always predict all the ramifications that would have. So oftentimes we're screening hundreds or thousands of different variants, right, around a central theme. We're knocking out different things, we're overexpressing at different levels, and we're trying to develop screening processes where we can pick out the winners, right, and take that kind of elite strain and kind of further characterize it down the road, right? And I'll show you some examples of that for some of my lab's work with psychedelics production here in just a few minutes. So now diving into kind of the section I'm calling psychedelic medicine, right? But before we talk about psychedelics, I want to talk about mental health, right? Because that's really the driver behind what we're doing, right? And I've broken down my kind of thoughts on mental health and mental health disorders kind of in 3 main categories. First, the mental health disorders are prevalent, right? If we look globally, there's about 1 in 8 people worldwide that have a diagnosable mental health condition at some point in their life. And that seems like a lot, right? That's 1 billion people, over 1 billion people, right? But if we look locally to the US, right, that number doubles, right? The rate of mental health disorders is twice the global average in the US. So that's 1 in 4. That's our children, our significant others, our family members, our friends, our coworkers. Everybody is cares about someone who is affected by a mental health disorder, right? So it's widespread, prevalent, and it's getting worse, right? With more global unrest and stresses in life, it's kind of driving some of these mental disorders to be more prevalent. And when we talk mental disorders, right, we're talking everything from addiction to depression, anxiety, post-traumatic stress, you name it. So those are all kind of lumped together in this statistic. But these things are also expensive, right? If you have a diagnosable mental health disorder, whether or not you're getting treatment, on average, you're going to live 10 years less than the kind of general population who does not have this diagnosable mental health disorder. So that's kind of a measurable kind of outcome on everyone's life. It's an impact. And that 10 years is like really close to the same statistic that you would have if you had a smoking addiction, right? So smoking causes you to live about 10 years less. And everybody understands that smoking is bad and connects the idea of smoking with a shorter lifespan. But that same connection is not as clear to everyone when we talk about kind of mental health disorders and mental health treatment, right? So any technologies we can have that help to kind of shorten that gap and make that can really improve kind of the length and quality of life for people, right? But it's also expensive in terms of money, right? The US, kind of the holistic cost of mental health treatment and losses due to kind of mental health symptoms cost the US about $190 billion a year, right? So that means the US is spending about the entire GDP of Greece, right, towards treating and managing mental health. And the losses due to, it's kind of a holistic number, losses due to like calling in sick from work because you're having a bad kind of mental health day. So they're expensive, so prevalent, expensive. And then, but the good news is they're also treatable, right? We have a lot of antidepressants. I'll use depression as the stand-in here. But we have a lot of antidepressants that work for a lot of people, right? Doesn't mean they're perfect, right? But many people find relief from their symptoms with treatment with mental health, with let's just say SSRIs for depression, Prozac, as an example, right? But the problem is about 24% of all the people that seek mental health care treatment would say that they have unmet mental health care needs, right? So the treatments that exist currently, pretty narrow in scope of mechanisms of action. They don't really work for a pretty large percentage of people, right? About a quarter of those. And there's even a larger percentage, right, that maybe wouldn't say they have unmet mental health care needs, but the side effects of their treatment that are helping their kind of mental health are causing other problems, right, that they'd prefer not to have. And there's room for improvement in kind of existing treatments for, let's just say, depression, right? But the exciting thing when we start to talk about psychedelic medicine is that people are starting to explore the use of kind of classical psychedelics, right? Psilocybin is the one that I'm going to focus on today, but there is many others, right? You've got DMT and mescaline, ibogaine, right? You name it. There are several other compounds out there that are being explored, most of which are natural products, not all. And there's derivatives there that are not natural products. But in 2018 and 2019, the FDA identified 2 clinical studies. So when I say clinical study, I mean studies in humans. That they kind of earmarked this fast track path called a breakthrough therapy designation, right? And that's effectively the FDA saying that the preliminary data from this study looks so promising that it could revolutionize kind of the treatment for, in this case, it was like treatment-resistant depression or treatment-resistant or like depression in kind of terminally ill patients, right? Those are some of the earliest trials that were going on, right? So they said for that population, this looks to be really promising. Right. And so that kind of gave the federal government stamp of approval, maybe not the whole government, but at least the FDA's wing. It said, there's some potential here. We should study these more. Right. And that's about the same about time when I started getting into my psychedelics research. Right. I started doing this work about 2017 when I started here at Miami. Right. So good news, bad news. In general, you should take away that mental health disorders are a big problem. Right. And the psychedelics have some potential to maybe help, right? So pivoting a little bit, I want to talk to people about what are psychedelics? Are they safe? Are they dangerous? Right? There's been a lot of propaganda shared over the last 50 years about psychedelics. So I like this plot, right? And let me explain to you what these axes mean. This y-axis is labeled dependence potential, right? You should think about that as like how addictive are these compounds, right? Things that are high in this rating are going to be things that are very addictive. Right, things that are low in this rating are going to be things that are not very addictive. And on the x-axis, right, think of this as safety. Things on the right are going to be things that are very not safe, right? And things on the left are going to be things that are very safe, right? So, and that's quantified by the ratio of the dose that it takes to feel the kind of impact or the effect of this drug over the dose that it would take to kill you, right? And as those get closer, the chances of overdose and kind of bad outcomes are greater, right? And these ratios, they change person to person, right? So depending on, so the further you can be away from, the lower that ratio can be, the better off you're going to be the more safe that drug's going to be, right? So looking at the data that's here, right? If we look into that top right corner, the things that are very addictive and very dangerous, those are going to be the kind of classic ones, right? If I ask people what drugs are dangerous and addictive, right, you're going to tell me things like morphine, cocaine, heroin, right? It's not on here, but methamphetamine would also be in that space. But not too far removed from those are drugs that many of you are probably using right now, right? Whether that's nicotine or caffeine or alcohol, right? I've always found it interesting that I could never draw a line between caffeine and alcohol without having roofies included. So I thought that was just an interesting observation. But more to the point of what we're trying to talk about here, if we look down in this kind of bottom left-hand corner, the compounds that out of this group selected, right, are the ones that are the most safe and the least likely to be addictive, are going to be the psychoactives, right? THC in the case of marijuana, LSD, and psilocybin or other tryptamines. So there's been a history, right? There's been a history of recreational use for these compounds that have shown them to be very safe, right? To have low abuse and harm potentials. And much of the propaganda about flashbacks and psychosis that's caused in kind of later life due to these compounds is just that propaganda. There's not a lot of science that backs up any of those claims, and really was kind of thrusted from the federal government on their kind of war on drugs they kind of initiated in the 70s. So pivoting towards some of the work from my lab and kind of what we do and the way we're tackling problems specifically. I'll first want to introduce to you some of the biosynthetic pathway, right? We haven't talked a lot about science yet. We're going to get a little deep in the data for a little while, and then I'll come back to kind of the higher level talk here in just a minute. But when we think about this pathway, right, we start our pathway towards the biosynthesis of psilocybin in E. coli, which is a little different than the way it's done in a mushroom, but many of the core principles are the same. What we do is we feed this compound here, this 4-hydroxyindole. It is a compound that's not naturally produced in E. coli, but it is cheap and widely available. It's something that we can just add into the fermentation broth. It's uptaken by the E. coli, and they can do biochemistry with it from there. And then that is condensed with naturally occurring serine using an enzyme that's the tryptophan synthase, right? That occurs naturally in all bacteria. What that does is it makes a non-natural amino acid, right? So this is 4-hydroxytryptophan, right? Where it would naturally make tryptophan, which is one of the 20 amino acids that kind of all bacteria produce. This is a non-natural version that has just this extra hydroxy group at the 4 position, right? And so that's using E. coli DNA to get to that stage, right? So from there, we took an enzyme from the magic mushroom, Psilocybe cubensis, otherwise, and it's the decarboxylase out of that enzyme, right? The Psi-D is the name of it. And what that does is that cleaves this carbon-carbon bond to release CO2, right? So it decarboxylates that compound into this 4-hydroxytryptamine compound. And this is the first concerted step towards our psychedelic compound. We also grabbed another enzyme from that same biosynthetic pathway, Psy-K, which is a kinase. And what that does is that uses ATP, right? One of the energy molecules that exist naturally in the cell, and the regeneration processes also exist naturally in the cell. And that phosphorylates that hydroxyl group at that 4 position, right? This also stabilizes the compound and creates a kind of good sink to drive the biosynthesis forward. This step's also really important because this is the step that makes my process more economical and more environmentally sustainable than traditional chemical synthetic processes, right? We can make these compounds kind of in a lab and using your organic chemistry skills. But doing phosphate chemistry is hard. It uses really toxic components, and it's really low yielding, increasing the environmental cost as well as kind of the financial cost, right? So having a really efficient step here is really important. And it's something that biology is really good at, right? They have natural phosphate donors in ATP that these enzymes have evolved to use. And then we have 2 steps, right, that are catalyzed by this Psi-M, right? And this Psi-M is a methyltransferase. So what it's doing is it's moving a methyl group onto this nitrogen at the terminal nitrogen here. So this methyl group is sequentially added twice, right? So we start with just an NH2. We then add a first methyl group to create biocystin and a second methyl group to create psilocybin, kind of our end product that we're interested in this process. And we find that this is, and I wouldn't be talking to you today if this didn't work, right? We pieced this together into a bacteria, right? We screened a few strains and we were able to find something that made the smallest amount of psilocybin you can imagine, right? We're talking like 2 milligrams per liter, right? 2 parts per million. So that's a really low concentration, but it worked, right? And once we can get it to work, that's where my lab thrives, right? I'm really good at manipulating these pathways to get it to work better, right? But first, I want to talk a little bit about the benefits of this approach, right? Once we've developed a strain that can kind of do this biochemistry, right, it's now DNA encoded and autocatalytic. What that means is that that strain exists, and I don't have to redo the chemistry again, right? I can just pull that strain out of the freezer, put it into the proper conditions, and it'll produce this compound kind of spontaneously without a ton of kind of prep work or intermediate purifications, right? It's also one pot, right? If you think about the chemical synthetic approach, right, there would be multi-step reactions. You'd have to purify and concentrate and manipulate each step individually as you go through the process. So it makes this process a lot faster. It also makes it a lot higher yielding because you have losses at every step when you have to manipulate things in that synthetic route, right? And then I hope we can all agree that this is way cooler, right, than organic chemistry or farming of mushrooms, right? The ability to kind of manipulate these organisms to give bacteria who's never seen psilocybin before the potential to manipulate those pathways is something that or to do the chemistry that the mushrooms can do is cool. But I'm a little biased, right? So I was given a few questions that came in at least a few days early of this through the registration process. So I wanted to kind of highlight a few of those as I go through the talk. The first question is why synthetic versus naturally derived compounds, right? And we can think synthetic as the chemical derivative or the biosynthetic process that my lab does. And so the idea there, and that opposed to kind of the kind of natural occurring kind of plant or fungus that you could, in theory, propagate. And I think the answer here lies in kind of the end use, right? Certainly people have been using psilocybin mushrooms for a long time, right, to kind of self-treat these applications. But as we pivot, right, towards trying to develop technologies that can help others, we want to be able to have a very reproducible product that is going to enable the approval of kind of governing bodies where these things could be prescribed to people that would help, right? And so the truth is, whether you're doing a chemical synthetic, a biosynthetic, or taking a naturally occurring product, you're going to get the same chemical structure, right? In the end, it's purified and it's a crystalline product that looks identical across all three of the kind of methods. But taking this kind of engineered approach allows us to scale things more reproducibly. It allows us to make a more standardized product that actually has a path towards approvals from the FDA level, so that compound can be prescribed to get to humans rather than kind of a recreational end use. Okay, so diving in to think about how my lab manipulates these pathways and how what kind of our bread and butter is, right? Once we've gotten this pathway to work, we've made this 2 milligrams per liter, right? That's, you see this box here says where we started, right? That's our 2 milligrams per liter level. So at that point, we had a DNA construct that we could manipulate and tweak, right? And so we can change the promoter strengths, which is what we did here. We could also change ribosome binding site strengths or promoter configurations, like where the promoters and terminators occur in the construct. And in this case, we made about 400 different variations on that kind of central core pathway. And we then tested their abilities to produce psilocybin, right? And again, we started at 2, but we were able to find that as we manipulated these, right, out of those 400, you can see there's lots of little bars stacked beside each other in this plot, right? We're able to find a select few kind of elite producers, right? Things that were well balanced, that this pathway was integrated into that complex web of metabolism more efficiently, right? So we're able to get to about 60 milligrams per liter just by tweaking the genetics behind how this pathway was expressed. Remember, it hasn't had millennia to be evolved to be well integrated into metabolism, right? We just did that on like a Wednesday afternoon, and by Sunday it was producing psilocybin. So we were able to kind of speed that up and tweak the expression of all these to kind of balance all of those cofactors and precursors and intermediates that we had, that I discussed in kind of the earlier case study. So once we had kind of tweaked the genetic optimization side of things, we went and looked at some of the fermentation optimization, right? There are some key process parameters that we can manipulate that can kind of help the cell make more or less product. So when we look at this particular case, this is just one snapshot of a piece of what we did, right? We looked at the supplements that we add to the media, right? We could also look at the temperature or dissolved oxygen levels or the types of vessels that things are grown in, right? Those are all valid options on the fermentation optimization side. But we looked at the concentration of that initial precursor that we add, right? Because it's a little toxic, and so adding too much is detrimental to the cells. And then we also looked at the concentrations of methionine and serine supplements that kind of help boost this pathway in particular's performance. And we're able to kind of manipulate those, and we're able to find some that gave us another kind of 2-3-fold change improvement over kind of our best, taking our elite mutants from the genetic screen and then rescreening under a variety of fermentation conditions. So we went from 2 milligrams per liter up to, let's say, 130 or so at this point. And so we're starting to get there, but really it's that gram per liter mark, right? We need to get to 1,000 milligrams per liter before things start to become industrially viable. Below that point, the kind of purification and downstream end applications are a little too expensive, right? So to give you a visual what this looks like, on the x-axis here, I have the intermediates of the pathway. So this is the starting substrate that we add, right? And this is the first, second, third, fourth intermediate until we get to our final product, psilocybin. So when we first start and we first get something to work, it looks something like this, right? We see that we fed in our intermediate, our initial substrate. A lot of it was consumed, right? But it got stuck in this first step of the pathway, indicating that the enzyme that converts this 4-hydroxytryptophan to the 4-hydroxytryptamine is kind of rate limiting, is struggling. But upon some genetic optimization, right, when we tweak the promoters and rebalance the pathway, we're able to see that we can now consume all of our starting material. We start to build up less of that first intermediate product, and we start to push our product or that mass through our system to get to that final psilocybin concentration that's a little bit higher, right? But then after we take that genetic elite mutant and tack on the fermentation condition optimization, you can see that we can kind of push that flux even further down the pathway, right? We're now kind of have no detectable levels of the first few intermediates, right? And we've started to push that flux closer and closer to be more and more towards our final product, right? So there's a lot of engineering principles at play at this, right? But visually, this is kind of what we're doing. We're trying to manipulate the genetics and conditions surrounding this cell to enable it to kind of make more and more of these kind of products. And once we've gotten to this point, once we've understood this strain in kind of a small scale, we've kind of run into limitations that are related to it being in small scale. So we go and put it in one of these, what's called a bioreactor, right? So it's like has a lot of autonomous control systems built in, and we can measure and control for pH and dissolved oxygen and foam. We can pump in extra oxygen, add different environments, those types of end applications. And what this also does is also mimics the way that these things would be produced in industry, right? All the industrial scale fermenters that would be not benchtop, but enormous, are going to give you this same set of control features. And once we went to this and we found that there were a few key parameters that we could manipulate further to kind of improve this kind of process, right? I'm an engineer. I think about process design a lot. We were able to kind of achieve titers that broke that 1 gram per liter kind of threshold. Right. And so here's kind of the raw data. And if we really just care about psilocybin here, which is the green, right, we can see that over the course of 3 days, we were able to improve that psilocybin titer to over 1 gram per liter of final product. Right. And again, that's that threshold, right? I said that 1 gram per liter for these high value compounds is what we've identified as kind of the point when we can start to think about industrial scale up, industrial viability. Okay. And so that all work, that was our initial work, it published in 2019 in this paper here. And this is really when my life changed forever. I started getting a lot of phone calls from press, lots of people interested in things that I was incredibly excited to talk to, right? Journals like Scientific American, Chemical Engineering News, NPR. These are the people that I've always admired, right? It's really cool to see some of my work focused and highlighted in some of these platforms, right? And then there was things like High Times and Mary Jane and Vice News that I never expected to cover any of my work, right? Although it kind of makes sense now that we're working towards psychedelics. And then some people just don't get it right. I don't know who Paul Seaburn is. I never talked to him, but I did not engineer bacteria that poops magic mushrooms, but I engineered a bacteria that uses the DNA from magic mushrooms to enable it to produce the psychedelic compound. So that's pretty cool. All right, moving forward, right? From there, right, I went to Reddit, right? Because what good scientist never cruises Reddit to see what they're saying about your work, right? And I found a lot of people that were interested in the recreational market for some of these compounds, and they want to know, like, can we invest in some of these it's lucrative technology, right? And then some people went a little further to kind of explain how to do it yourself, which I would argue is a little misguided. But I took this as kind of a funny thing, right? It said, plus in a week or two of running even a small reactor at 1 gram per liter would put you in extreme surplus of product and money. I've been doing this for 5 years now. I can't say that I'm in the extreme surplus of product and money just yet, but I like the interest. But this led me to the next problem, right, that I was interested in, right? It's like, how do we regulate this, right? These compounds are regulated by the DEA. I have a DEA Schedule 1 license, which comes with a heavy burden of paperwork to be able to manage my lab and make sure that I can continue to do the work that I do. But I wondered, right, if my strain was to get out of the lab, right, could it be used in the same way that yeast is used for homebrewing beer? So I got a team of a staff scientist that kind of managed a team of pretty new undergraduates. And so I tasked them with this idea, right? Under the regulated conditions of my lab, can we like mimic what somebody would have done in their garage, right? So we ordered a sous vide that you would use for like preparing meat off of Amazon, right? We had kind of a water bath that we used to kind of keep the temperature control. We used aquarium pumps to pump in oxygen into a bottle with just kind of a stir bar. And surprisingly, right, it worked pretty well, right? We were able to achieve greater than 300 milligrams per liter. Granted, about 3 times less than what we got in our like $50,000 bioreactor. But we were able to assemble this for just a few $100, right? And that told me that like maybe we should think about the regulatory considerations. We ended up writing a paper, not a how-to homebrew your own psilocybin, but to kind of highlight some of the needs for regulatory considerations in this kind of new synthetic biology world of drug production. And so that's some kind of an interesting project that kind of I involved staff scientists and undergraduates in my lab. And to cap it all off, we did all this, and it only costs about 2% of what it would cost to get the equivalent amount of drug in the street value of mushrooms, which is kind of wild, right? That we could develop this technology that would be so quickly cost competitive with something that's existed for decades. But then I'm going to pivot, right? And my lab's now going to kind of start to pivot towards other natural compounds. Watching the time. All right. So mushrooms don't just make psilocybin, these psilocybin producing mushrooms, right? There's this whole biosynthetic pathway, and all of those other intermediates, all those other metabolites are also present, right? And there's probably a difference, right, between taking a purified psilocybin version, right, and a mixture of these metabolites that would exist in the kind of natural raw product like the dried mushroom that recreational users would take. And I was interested in exploring those, right, with the idea that maybe some of these compounds could be better drugs, right, than psilocybin, right? Because psilocybin has this side effect of a 6-hour hallucinogenic experience, right? That's not ideal for a depression therapy, right? But the problem at the time is that nobody could study these compounds because there was no commercial availability. Now, a few companies have started to provide these to the tune of like $100 a milligram, which is a little bit crazy, kind of prohibitive for any type of real study. So I set out to kind of make psilocybin or E. coli strains that could produce these compounds so that I could make them available to researchers. Right. So to kind of draw some parallels with our psilocybin pathway, we first started out thinking about norbaecistin, which is one of the intermediates that we're interested in. And we can get there by just eliminating one enzymatic step in the biosynthetic pathway. So instead of expressing psi D, K, and M, we're now just expressing psi D and psi K. And we're able to take a very equivalent process for the genetic optimization, right? Here's the data you saw earlier with our psilocybin pathway. We're able to find that our norbaecistin pathway worked a lot better. There's a lot of reasons for that. It's a simpler pathway. There's less burden, less cofactors needed, etc. And in the end, once we scaled up in the bioreactor, we're able to see similar results. This time we got to about 1.5 grams per liter for norbaecistin. which is a really cool compound. And I'll explain more about that when we get into some animal studies. But we also are continually trying to improve some of our psilocybin biosynthesis technology. Right. And so this is a project that's currently ongoing in the lab. It's not published yet, but we are in the process of working on that. 2 excellent students were involved in that project, graduate and undergraduate students. But the idea here is that we've done all this work to date, made really high producing strains, E. coli strains for psilocybin production, but we have only used one organism, right? We only use psilocybe cubensis DNA, right? And who's to say that when we're plucking DNA from a mushroom and sticking it into a bacteria, that the best solution is like the first one that we come to? So what we did is we went to the literature and we found 3 other psilocybin producing mushrooms. There's about 200 in existence, and there's more and more data coming out every day with these genetic sequences. But at the time when we started, that was really the only ones we could find. And so we looked at these other 3, we pulled their pathways, and we evaluated whether the enzymes that are in those pathways, the equivalent D, Ks, and Ms from these other organisms, could work better in our bacteria. And long story short, we found that turns out there are some better options there. Much of what we were working with was like the cubensis one is pretty good, right? Obviously, we've seen good data from that. But by manipulating this one gene from this Gymnopilus dilepis organism, we were able to develop a new strain that is able to produce about 1.5 grams per liter of psilocybin, which is a significant improvement over our 1.2 that we got with our original kind of case study back in 2019. So this is now the highest, the most productive E. coli for producing psilocybin in the world. I would say the most productive recombinant organism for producing psilocybin in the world. There's also a few other key kind of process-oriented things that are going to make this actually a much more economically viable production route over our kind of current best published route. So this is some really exciting data. I'm looking forward to getting this paper out in the next 6 months or so. But pivoting towards some kind of new work in my lab, we're working towards a whole variety of different derivative products now, with the idea that maybe the kind of psilocybin compound is a good start, but it's got some drawbacks, right? The hallucinogenic side effects, for one, that make it maybe not the best drug end product. So we're working with the kind of existing pathway, and we had to expand on it a little bit to add a few extra enzymes for some extra functionality. But now we can feed not only 4-hydroxyindole, which is where we started previously, but we can feed a whole slew of different indoles that are derivatized at any position that we want, right? We can add chlorine groups, fluorine groups, and nitro groups, and amine groups, right? To try to make new compounds that might have interesting kind of pharmacology and biochemistry and applications. And so this is work that was led by staff scientist in my lab, Bill Gibbons, and a undergraduate, now master's student, Jessica Flower. And I'm running out of time a little bit, so I want to get to some of the exciting stuff at the end. So I'm not going to go through all the details here. But fundamentally, we screened 49 different varieties, 49 different inputs, right? 49 different substrates. And in the end, we made 13 different psilocybin derivatives, right? And these are a lot of halogenated derivatives, right? Like 6 and 7 iodo-psilocybin, or 5-fluoro-psilocybin. I guess we have 5, 6, and 7 fluoro-psilocybin, methoxys, and methyls, right? So these are all new compounds that are built off of a central psilocybin core that likely have some interesting bioactivities, but might have a different experience or a different kind of drug-like property that we're going to start to now screen in a variety of different end applications. So that's really exciting. I see that as a future direction of my lab, and some of the preliminary data from a collaborator will explain why I'm so excited about this direction here in just a few minutes. Right. And then I think the last science before I jump into some of the animal studies I want to talk about is some work from a master student in my lab, Lucas Friedberg. This work was published last summer, where we're not only making psilocybin, but we can also make DMT, right? And DMT derivatives, right? Which is another natural hallucinogenic, right? That is made into a tea called ayahuasca, which is kind of common in kind of South American ceremonial uses. But if we think about a derivative product called 5-methoxy-DMT, right, which is just a methoxy group different from DMT, it's the compound that's found in the kind of gland excretions from the Colorado river toad, right? And that's frequently smoked and used as kind of a very fast-acting psychedelic, right? So instead of a 6-hour psychedelic experience, right, this compound in particular gives about a 15-minute psychedelic experience, right, which makes it an interesting kind of drug product for some reasons and not so much for others. But we're able to piece this pathway together. In the end, we're able to achieve about 100 milligrams per liter of DMT production, so lower production than what we had seen in our psilocybin case. And there's some reasons for that. And we're still working on a few of the details. But what's interesting about this is that we compiled DNA from all walks of life, right? We have a E. coli's native tryptophan synthase. We have the magic mushroom decarboxylase in the pathway. And then we pulled a methyltransferase from humans, right? So something that's naturally expressed in the human body, particularly in the human brain. And that was able to biosynthesize some of these DMT derivatives. We've also started to think about feeding in different compounds, similar to what we did in the psilocybin case study. And we're starting to see some really cool kind of derivative products that we can make here, and we're working on scaling some of this up for some animal testing in the future. So that's something that I think is really cool and some work I wanted to highlight. So similar theme, slightly different compound. Great. So question from the audience: does this ability to engineer psychedelic medicines in this manner make it more likely that it will become legalized for mental health therapy? So I kind of highlighted that earlier, right? I think that trying to say that we can prescribe someone a mushroom that could be very variable in its kind of potency, it poses issues when we think about kind of legalizing or prescribing some of these compounds in the long term. So I think this more rigorous kind of chemical synthetic or biosynthetic is certainly necessary for that. And I think the biosynthetic poses some economic value that kind of can supersede in at least some areas the chemical synthetic. And then what is the possibility on timelines for legalization? I'm not an expert in this area, but I do follow the field pretty closely. I think within the next year, we're probably going to see MDMA, which is ecstasy, being approved for certain kind of psychedelic, for certain end applications or end diseases. That has its side effects and it's not ideal. And then psilocybin is probably the next one in line. So I would say psilocybin therapies will probably be approved in some form or fashion in the next like 2 or 3 years, where you could kind of get a prescription for this. Although there are some big kind of legal directions that we have to kind of figure out from there. But so thinking about some of the preclinical data in the future, there's some pretty interesting work that's being done by a collaborator of mine, Matt McMurray. He was at Miami until this last year, but he has kind of been one of my earliest collaborators in this work. And so most of the data I'm going to show today is going to be from his lab, but I have a whole range of collaborators that are just now getting started studying these compounds, right? So head twitch response is how we think about a rodent being, it's how we measure whether the rodents are seeing kind of a hallucinogenic experience, right? So we can give rodents known hallucinogens and they will shake their head in a fashion that is more frequent on a dose-dependent kind of response to these hallucinogens, right? And the way that we do this is we do a small surgery to affix a magnet to the top of their head, and then they're tested in kind of a container that has a wrap of wire around the outside, and that magnetic field within that kind of coil of wire gives a signal that we can then process electronically on that outside. But you can also visualize these kind of with these video recordings. And Molly, if you can cue up that video, I want to show you what we're kind of trying to quantify in this kind of end space. And so we can see a rat here in this head twitch container, and you're going to see a couple head twitches. One right there, you saw that like quick little head twitch, and then another one right as it was coming down. And so that is difficult to quantify, right? But we're starting to develop kind of hands-free, kind of technology-enabled solutions to be able to quantify some of this, right? And again, this is the best thing we have to kind of think about, is this compound hallucinogenic in this rodent? So as we kind of go to some data, right, we started to test all of these compounds we're making, right? We now are making all 4 of the compounds that I had highlighted as also exist in the natural mushroom. And when we think about increasing the dose of these compounds, if they're hallucinogenic, we should see a rise in this kind of head twitch response, or the number of head twitches, right? And so in psilocybin, we see this nice peak around 1 mg per kg. But in Biosynthesis, Norbiosystin, and Oregonacin, we don't see a hallucinogenic response, even though they're very structurally similar to psilocybin, which we found was interesting. So that's not necessarily a bad thing, and I'll explain more about that here in just a minute. And then introducing some kind of new data on head twitches, right? There was these articles that came out over the last week, which I think is interesting, right? That really talked about kind of the female experience with magic mushrooms, and how there's this growing trend of working moms using microdosing mushrooms for to kind of help with their kind of day-to-day tasks and make them focused and those types of things. And I'll be the first to say that microdosing, the rigorous science clinical studies are very inconclusive and don't really show that microdosing does much, right? But the placebo effect is important, right? And I think there's some interesting kind of end applications, right? And this has come out in the last week, including a nice article in the Wall Street Journal that you could look up. But where I'm going with this is that these compounds have not been studied all that much in female models, right? Most of the time, drugs are studied in males only, because there's kind of extra hormonal cycles and things in females that kind of make it difficult to study. But the truth is, these mental disorders are more prevalent in women, right? And that if we don't study it in women, we're going to have we're missing kind of a large population that could have interesting side effects or bad side effects that we need to be able to manage and understand. So here's a study where we started to look at not only women, but also adolescent males and females. So kind of mimicking children. And there's some really cool data here with the adolescents. So early and late adolescents, for both males and females, we see a really reduced production of really reduced head twitch response, right? Which maybe doesn't mean that the children are not hallucinating, but it means that there's something there. Maybe the head twitch is a different frequency or has a different intensity, and we don't really, it's harder to quantify. So there's some unknowns there that we have to figure out. But interesting here from this bottom data, right? What we see is that the female, these are all done in rats. The female rats, right, in the proestrus stage have a dose response or head twitch response that looks very similar to that of males. But in the diestrus stage, right? So proestrus is like, it'd be the equivalent of kind of at the beginning, like at the beginning of a cycle versus the end of a cycle. The kind of diestrus stage, we see an elevated head twitch response, right? Both in kind of baseline as well as in the kind of with the drug kind of applied. So that's really interesting. We don't really know why, but there's something about kind of these natural cycles that maybe changes kind of the perception of these drugs, or how at least kind of this physiological response in these rodents, right? And it really illustrates that we don't know everything that we need to know, but there's some interesting kind of future study here. And in both cases, right, we also looked at the concentrations of the serotonin receptors that these drugs act on, and that was pretty constant across the board. So there's more to the story is really what that is, right? So preliminary data, but some really cool kind of outcomes that we need to explore more. We also have looked at kind of sucrose preference testing, which the idea here is that if like a rodent like prefers sugar water, just like humans, right? But a depressed rodent kind of doesn't have a preference for sugar or normal water, right? And so if you can give a rodent an antidepressant, a depressed rodent an antidepressant, right, and they become less depressed, right, they'll start to kind of transition their preferences more towards the sugar water. And you can kind of quantify that by the amount of water they drink or their ratio between the two. And so what we can see here is that we can see that this is a test where kind of the natural antidepressant, SSRI, fluoxetine, the active ingredient Prozac, it fails this test, right? It's known to fail this test. So all these tests aren't perfect. But what we do see is we see this reversal with psilocybin, which was kind of expected based on kind of clinical evidence. But norbiosystin is even more exciting, right? Because remember, that's the one that's non-psychedelic, has no hallucinogens, no hallucinogenic properties, at least in the rodents, as far as we know. But we're seeing this kind of antidepressant activity, right? Which is starting to kind of tease at the idea is, do we need hallucinogens for this end application. Right. And then we've also looked at a test called the forced swim test. Apologize for moving through this quickly. I'm trying to finish up within an hour. And these animals, so the idea is we put these animals down in an inescapable chamber, right? They're treading water, right? A depressed animal kind of gives up faster and just kind of floats there, where a less depressed animal is going to actively try to escape more frequently, right? And so we quantify this by thinking about the amount of time that they're just kind of laying there. How much time are they immobile? And you'll find in the vehicle, right, this value is high, right? But in several of our drugs, norbaecysteine and psilocybin, as well as fluoxetine, again, the positive control Prozac equivalent, we can see that there is a reduced immobility time, indicating some antidepressant action. So that's something that we are excited about, right? And we're particularly excited about this idea that this non-hallucinogenic compound could be having some antidepressant side effects, which really lowers the bar and makes this idea that we could approve this compound and that it could be more economically viable, I think, pretty high. Right. I'm going to jump past this question for just a second to talk a little bit about what I see as the future of mental health treatment, and then we'll wrap it up and have some time for questions at the end. When I think about future mental health treatment, I have to think that we're going to find better drugs than what exists today, right? I firmly believe that there's going to be a fast-acting, psychedelic-inspired, but probably non-hallucinogenic kind of standard of care for these compounds, right? And the reason for if it's a hallucinogenic, especially like a psilocybin level one, right, where you have a 6-hour hallucinogenic experience, the cost of treatment is going to be astronomical, right? Because you have to have a kind of monitored environment, and you have to have paid shamans or psychiatrists present to kind of help manage that hallucinogenic experience. And if we can understand how these mechanisms work in a more an easier to prescribe kind of format, I think that's really got to be the future. I think the idea that you need a daily dose and it has to build up in your system over weeks, like it is with our current kind of SSRIs, I think that's got to go, right? And I think psychedelics pose a ability to have monthly or quarterly type doses to have this antidepressant effect. And that's really important because these mental illnesses, one of the side effects is that it makes it hard to remember to take your doses every day. And if you don't take them every day, they don't work as well, right? So that's something that's kind of in conflict and something that I think the psychedelic medicine can help. I had this maybe far-fetched idea that there might be a future where since the side effects are so low for these compounds, that there might be some prophylactic applications of these systems, right? Maybe you've gone through a traumatic experience, whether that was a bad car crash or the spontaneous loss of a loved one, or a traumatic like birth experience, right? That prophylactically they could say, you should take this kind of psychedelic inspired non-hallucinogenic tryptamine to kind of fend off the likelihood that you're going to have depression or post-traumatic stress disorder in the future, right? So I could see that. I could also see tailored drug therapies, right? Where we think about maybe there's a psilocybin partnered with a non-hallucinogenic compound in some combination that kind of could be tailored to the specific mental health disorder that you have, whether that's depression or post-traumatic stress or addiction. So I think there's some really cool work that we could do with some of these compounds that we're doing. And as we learn more about these diseases, we can tailor those kind of doses and chemicals to their specific end applications, which is something that's not really being done now. So that's my future, what I see in the for mental health treatment. When I think about the future of my lab, right, I'm really focused on the biosynthetic side of things, right? So I'm looking to kind of always expand these pathways and make new diversity of kind of drug candidates that we can then produce. And I'm really focused on the process, right? Can we scale this up? Can we make enough of this purified compound to give it to collaborators to test? And I'm really looking to expand the impact of this work beyond kind of the metabolic engineering sphere, but I'm trying to build as many collaborations, right? That's my goal for 2024, is to build as many collaborations as I can with like-minded researchers across the country that want to study some of these compounds that we're making, right? To think about how it affects their model, right? Whether that's kind of neurodegeneration or addiction or some type of other interesting kind of anti-inflammatory property that these things might have, right? I'm really trying to look at for a holistic picture of people in medical schools, neuroscientists, biochemists. They can kind of build a diversity around some of this work so we can study these and learn more about them. That's something that really gets me excited. I love those conversations I have with people. hopefully you're asking yourself, how can we support this work? And it wouldn't be part of this is me sharing what I'm doing, and I'm very excited about it, but also I work with students, and that's my goal. And it takes, oftentimes there are things that we want to do that we can't do because we're short on money, right? And I think specifically funds that can help support the undergraduate trainees in my lab is something that I frequently need, right? So if you're interested in this, you say, I want to get involved, right? And I'd love to support undergraduates that are doing undergraduate research, right? Or I would like for him to be able to incentivize more collaborators to study some of his drugs with some kind of preliminary kind of study funding, right? Or you want to see more animal study work, or you want to give students an experience to go share this research with kind of the outside world at a conference or something like that, right? Those are all great end applications, and I would love to talk to you more about kind of what that could look like. There certainly are tax deductible ways for you to donate money that is earmarked specifically for what you want to see it work with. And if you want to do that through my lab, that's fantastic. You can email me. My email is pretty easy, jones@miamioh.edu, or Molly Young, which was the contact for this kind of event. And we can get you connected with advancement and make that happen. So I'd really appreciate anything you want to do to help support this work. And I'm really excited about the future that this is. And I've run a little over time, but I do want to end on the acknowledgement slide again, right? Again, remember, it's not me that's doing all the pipetting and moving and manipulating, right? But it's this fantastic group of students, staff, and collaborators that are kind of making this possible, right? This is a big team effort, and we're doing some really cool work. I'm looking to I've always had a very large kind of research footprint, research lab here at Miami. I try to impact students. That's why I'm at Miami. That's what makes me the most excited, that ability to kind of take a student that really doesn't know what they're doing, right, and train them and see them kind of grow as a scientist, grow as a critical thinker, and then go out in the world and do kind of big, impactful things, right? That's why I'm in academia. And my collaborators share a very similar thing. So with that, I don't know if we have a lot of time for questions, but I'm happy to answer them, and you can always follow up with emails. As anything, I talked a little longer than I planned on.
Yeah, we have had a few come in, and I don't think we'll be able to get to all of them, though I do believe that you have addressed some of the questions that came in. So I know you're not an anthropologist or a historian, but what impact would you say the stigma of drugs and drug use has on your work and on your research? Like those people who are conditioned to think of mushrooms as just hippie stuff, or those people who think about the chemistry of creating drugs and they go straight to Walter White and Breaking Bad, as far as, funding, respect, collaborators in general. What's the impact like for you?
Yeah, I mean, it certainly creates friction. I think for one thing, I think things are changing. I think public perception of psychedelics is not what it used to be, right? I think people are becoming more accepting and more happy, right? I across the board, I hear a lot of support for our work, 10 times more than I hear somebody saying like, why are you doing that work? There's no way this is just things, right? So that's exciting. And that's what keeps me going. Where it causes the biggest issues is like the structural hurdles that have been put in place, particularly at the federal level and the state level in many spaces, right? The fact that I have to have like a DEA license and manage all of and control and keep track of everything that happens like in a very kind of meticulous detail adds extra burden and takes away time that I could be spending doing other things, right? The ability, so the federal government has kind of classified these compounds as like not good for anything, right? That's kind of the category. And because of that, that's kind of limited federal funding for research in this area, right? So a lot of like most of the work that I've presented today was all funded by private industry. And so there's trade-offs there, and like funding directors are like the NIH is starting to think about funding more psychedelic work, and that's really good to see. But if I was making a biofuel, it would be much easier to find federal dollars, right? And so there's kind of structural things that have been put in place due to kind of how these compounds have been legally treated in the past that really make my life a little more difficult, right? It also makes like decisions at the university harder, right? Because the university has to balance. Can we have this public perception?
Right, we're a public institution. It makes sense.
Yeah, absolutely. And then
so one last question for you, or sort of a comment to invite comment. I really appreciated your acknowledgement of the lack of testing psilocybin and similar drugs on females. As someone who daily relies on Prozac for depression and anxiety. And also, I was thinking about, the women who are microdosing on mushrooms for focus. Well, I am really glad that ADHD medications exist, because as a adult diagnosed person with ADHD, it's like those things are so important. And I wonder how would those medications be even better if women were considered more in research and testing. It made me think of the recent reporting on how menstrual products weren't tested until recently with actual menstrual blood, and the impact of women in science on research. And so I'm wondering about women in your field of work, and are you seeing more interest from female students? And is this part of the Women in STEM movement, do you think, moving forward?
Yeah, I mean, I think anecdotally, at least, my lab is more women than men, right? Which is weird because I'm in an engineering
Awesome.
college, right? And some of my most
Right.
successful students
Yeah.
have been women. And if you saw the pictures on all the slides, right? Like,
hmm
and so I think that women are attracted to this. I think
Yeah.
it really could be because women are more kind of are diagnosed with depression more frequently than men.
hmm
On
Well,
the
and I think of, the issues folks have with SSRIs during pregnancy. If you were able to, come up with something that could help maintain a woman's mental health throughout the time of being pregnant, that's
yeah,
another
and
possibility.
studying the impacts that could have on
Sure.
the fetus and things that's a big
hmm
question.
Yeah.
There is a lot of good here, right? The NIH has really addressed this directly. Like they've effectively
hmm
said, we're not funding animal work unless you use
hmm
sex as a biological variable, right? They effectively say
hmm
you must test on men and women if you want this money.
Right.
And so that's been established in the last few years, and that's like really
hmm
driving kind of this forward. And so I don't
hmm
think we'll have as many issues.
That's
But
great.
from the scientist side of things, right, there's limited money, right? You can only run so many
hmm
experiments, right? And if you're going to
hmm
double everything, and arguably you have to more than double because you're trying to factor for these kind of hormonal cycles, and you might have to have multiple kind of study groups in women, right? It makes it hard, right? With limited funds, right? And you want to get some preliminary data. Sometimes it's easier to run that initial study in
I
men
would
to
say
see if
go with
something
just
there.
test on women.
Yeah, no, I think there's lots to consider
Yeah,
there, and
you have to just choose
it's
one.
no, it's inevitable that
Yeah.
you have to consider that now, and there's no way that you're getting a drug approved at this point if you've only tested in men
Right.
the whole way.
Yeah, for sure.
So
Well, thank you so much, Andrew, Dr. Jones, for being here today. This has been incredibly informative. And for those of you watching live or post recording, please feel free to reach out to Dr. Jones with any questions that you have, and let us know if you're interested in supporting his work. Thanks for being here. We will have another webinar for you tomorrow to continue Winter College Week. And we look forward to seeing you all online again soon. Love and honor.
bye. Thanks for all you attending.
Thanks.